Next keynote speaker is Vesna Pajic, bioinformatics analyst, team lead at Valsera. Strong education professional with a PhD focused in computer science. She is experienced both in academia and the biotechnology industry. In the last 15 years and more, next-generation sequencing technology is being developed constantly to become a powerful tool for getting insight into different biological processes within cells, tissues, and organisms. The data coming from NGS has all properties of big data. In this talk, she will cover basic ML techniques used for their processing. Vesna, the stage is yours.
Thank you, and thank you all for coming here. So my name is Vesna Pajic. I am a manager in bioinformatics in company Valsera, and today I will speak about machine learning techniques for omics data analysis. So I will first introduce our team, and then I will give a short overview of omics technologies, what are they and what are they used for. I will also present some omics data, just so you can see what do they look like and how we process them. And then I will speak a little bit about machine learning algorithms that are commonly used in processing these data. At the end, I will present one real case scenario from one of the recent projects we had, where our team applied these machine learning algorithms to help one of our clients. And hopefully we will have some time for discussion, but if not, I will be here all day, so you can reach out to me later. So Valsera is an international company. It is founded in January this year, and practically it is formed out of three companies, Eugentech, Pierian, and Seven Bridges, so all three very well-established companies before January. We are at Valsera focused on powering precision medicine and improving human health in every aspect. We have a very large team, around 600 employees all over the world, in Serbia, in US, UK, Belgium, India, Turkey. And here in Belgrade, we have a great bioinformatics team. A lot of members are here as well. We have more than 40 team members there, and it is a very diverse team. We have people with different backgrounds in computer science, engineering, mathematics, statistics, but also molecular biology, pharmacy, chemistry, and I think even more, not so sure. So I will first start by introducing omics technologies and what they are. Practically, we use omics technologies for exploring different aspects of living organisms, including human health, at which we are focused in Valsera. And we do that by exploring genetic material. So, as you know, there are DNA molecules present in every cell of our organisms. There are also other genetic material present there as well, but DNA is seen something as a recipe for life. So it contains instructions for growth, for development, functioning, reproduction of cells and of organism as a whole. So from the moment it was discovered, humans are trying to understand that code, to somehow see what is written there in order to understand different aspects of life, such as inheritance mechanisms, human origins, diseases, how do they develop, what causes them, then how to treat those diseases, how to design drugs, and so on. There is applications as well in agriculture and in environmental sciences and so on. So, as I said, in a cell, besides DNA molecules, there are other sets of molecules which are also very important for functioning of cells and organisms. And in life sciences, we are using some specific terminology for those sets of molecules. So the complete DNA material in a cell or in an organism is called genome. RNA molecules, mRNA molecules present in a cell at one moment are called transcriptome. Then we have proteome as a set of proteins present at one point in a cell. And we also have other omes like metabolome, epigenome, and so on. For each of these ohms, let's imagine them as a set of molecules in a cell, we have technologies to digitalize them and to explore them in some way. And these technologies are called omics technologies. So for genome, we have genomics, for transcriptome, we have transcriptomics, and so on. They are not the same. They have different ways of how they digitalize those molecules. And the data coming from those technologies is not the same. But they do share some common characteristics. And they are usually explored in parallel. So if you are interested in one biological phenomenon, you would probably want to know to explore not just genomics, but maybe genomics or, let's say, proteomics and transcriptomics in one, from one sample or from a set of samples. And that is why we think of them as one set of technologies called omics technologies, because they are not the same. From now on, I will focus on genomics and transcriptomics. So these are technologies for reading DNA and RNA molecules. But if you're interested in any of other of these, we can talk later. So how these omics technologies work? Well, they are practically based on the structure of DNA molecules. And you probably know that, but DNA is practically a polymer consisting of two chains of nucleotide, which they are connected together in a double helix structure. And each of these chains, or strands as we call them, contains sequences of these nucleotides, and there are 4 of them in DNA. We have adenine, thymine, cytosine, and guanine. RNA has similar structure, but it is not, it's only one strand, and instead of thymine, we have uracil in RNA. These sequences of nucleotides are quite long. For example, in human genome, we have around 3 billion of pairs of those nucleotides. So very, let's say, complex structure, but on the other hand, we have only 4 bases in this structure that we are interested in when we are working in genomics. So because of that structure, there is a way that how we can read those elements of a DNA. It is technology that has been being developed for a while. There are all 3 generations of these so-called sequencers. So this sequencing technology is technology with which we are reading those DNA molecules. And here is a picture, just an illustration from Illumina's website. Illumina is one of the most commonly used sequencer providers, and they are developing this technology intensively. They have different machines, but they all work similarly. They just have a different scale or different throughput. I will not go into details of all these steps. It is a very complex process, how we are getting from genomic DNA, so from biological samples, molecules, to sequences of characters which corresponds to those nucleotide bases. Very complex process. But what is important here to understand is that each step of this process can introduce some artifacts, some errors, some biases in our data. So this technology is not perfect. There is a lot of things that can go wrong. And all these errors will be contained in our final data. And this is something that we work with, and we need to account for these errors in our analysis. One important thing here to remember is that this genomic DNA, which is practically very large molecule, is first getting cut into smaller fragments, and these fragments is something that we are getting at the end as a result. So we will never get a whole molecule, but just these small fragments of text. So the output from a sequencer, after we prepared our samples and run sequencing, and the output will be practically a file. It could be one or several files. There are different formats, but most common format is FASTQ file. Practically, it is a textual file containing those reads that correspond to DNA fragments. And together with those reads, it contains qualities of the bases, how the sequencers estimate the quality of one base. Here you have just a few first lines, maybe 3 reads from the beginning of one FASTQ file, but if you can imagine, FASTQ file is a file with billions of such lines, very huge files, having sometimes several hundreds of gigabytes, and also containing all those errors that I have been talking about. So very complex data, complicated for manipulation, for processing. So this is our starting point where we start our analysis, and we call that raw data. From that raw data, we first take some common steps in the analysis. The first thing is some kind of pre-processing, because you don't want to have bases which are read with poor quality. You are not certain if this is the right base, so you would probably want to trim those out, or you may want to filter out all the complete reads. For example, if the whole read has low quality. But after that, we are taking some common steps, which are called secondary analysis. And these are assembly, alignment, variant calling, and gene expression analysis. Depending on whether you are sequencing DNA or RNA, you will use either variant calling or gene expression analysis. But assembly is a process through which you're trying to reconstruct your original genome. So as I said, the original genome has 3 billion of pairs, and here we have only billions of small fragments, so we are trying to reconstruct the original sequence. It is a very complex process. Practically, you start by overlapping those reads and then trying to construct these long sequences and to see how the original molecule look like. And this is one way you can go. The other way is when you know how this genome should look like, because we all share a very common genetic material. Our genomes are quite similar, so we know how human genome should look like. Then you can use this information and just try to map your reads to that reference genome. If you do that, this is called an alignment. So these are two ways where you can go from FASTQ reads. We usually do the second one, but of course there are applications where we want to construct the original genome from the start. And when you do this alignment, when you see the positions of reads, you can detect some differences. So for example, in reference genome, you may have a T in one position, but in a genome you sequenced, you may have some other base, like here we have cytosine on this picture. There is a process to determine if this difference is a real difference in genome, or it is something that is introduced during the sequencing. And this process is called variant calling. And it is done usually when we have DNA sequences. If we are sequencing RNA, then we will probably be more interested in understanding how many reads we have from that one position, because in that way we may know how much some gene is expressed. Is it active or not in one set of samples? And this is this gene expression analysis process, which we do in this part of the analysis. As an output from this secondary analysis, we usually have, these are just an example of one VCF file, which may be a result from variant calling step. And on the next picture, you have a feature count matrix or gene count matrix, which may be result from gene expression analysis. In the first VCF file, you have one row, one entry, one line for each of the detected variant. And in feature count matrices, you have for each, in rows, you have genes, and in columns, you have different samples, and numbers represent some relative counts, how much this particular gene is expressed in that particular sample. So these are outputs that we have after secondary analysis. So we first have FASTQ files with raw reads, then we try to give them some meaning in this way. But from there, we also need something to understand, okay, if we have difference, let's say, in this, if one of these genes is in set of samples, is that difference statistically significant? Is it something that we expect to see, or is it there just by chance? And this is what we do in steps that are called tertiary analysis. So we are trying to understand these differences and to get some insight into biological phenomena of interest. There are many of them. I here put just some five, the first five that came into my mind, but there are many more. Practically, on these plots, you have some visualization of these results, but the resulting files are also some statistics, metrics, and things like that. The plot, Manhattan plot at the top row shows it could be a result from GWAS studies. So when you are trying to analyze VCF files from a large cohort of patients or samples, then you would be interested in some locations in genome where you expect to see some variants. The figure in the bottom left corner is a picture which we can get from differential expression analysis. That is the analysis when we are comparing to gene expressions from different set of samples. For example, we can have normal and cancer cells, or we can have untreated and treated cells, and we want to see if there is difference in gene expression. Then we would do differential expression analysis. The third picture is from a single cell data analysis, where we want to somehow see similar groups of cells within one cell, or within one tissue. When I say similar, I mean similar by gene expression, which have similar gene expression. So these are just examples of the things that you may want to do after the secondary analysis, but it really depends on what are you researching, and there are many more possible applications. So where does machine learning fits in this process? Where I would say it fits in almost each of these steps. If you have in mind how complex the data is and how big the files is, then you know that we need some algorithms which can handle such data. The analysis is very computationally demanded, so we need machine learning can solve some of the problems. Here are some examples for secondary steps. Steps in secondary analysis: assembly, alignment, variant calling. There are machine learning techniques that are used in these steps, but I would like to add here that practically these algorithms for alignment, assembly, variant calling are pretty much standardized nowadays. So we at Valsera almost never had a client who came to us and said, let's improve alignment for me. So our clients expect these algorithms to work well already. And although they include some machine learning in them, in industry practically we are not improving them more. There is a room for improvement, of course, but there are already some best practices, some guidelines how to do alignment, how to do assembly, and this is somehow taken for granted. So we are not, at Valsera, we are not working on these algorithms. We are just using the ones that are already accepted in community. What do we do there, and where we can present all our creativity and expertise is in tertiary analysis. Here is just one example from a recent study from NIH colleagues, where they tried to show where machine learning algorithms are used, and they put these pictures so nicely and divided based on classification of machine learning algorithms. But you can see that almost every type of machine learning algorithm has some application in tertiary analysis. And this is something where we have more creativity with this data. I mean, this is technology which is still developing and still is being adapted by some new researchers. So there are constantly new applications occurring, and we have a possibility and chance to do something new. And instead of speaking about someone else's work, we decided, with agreement with my colleagues, I decided to present one of the recent projects that we did for one of our clients. So this is a real project that we had. This is our beautiful team and clever team that has been working on that project. Nicole and Voy are here. And beside them, we also have Neven on board and Jeff as a scientific partners. But practically, Nicole, Voy, and Neven did all the work. And the client came to us. They had been developing oncology drug targeting some metabolic pathways in cancer cells in order to kill them. And they were interested in predicting cell lines that are sensitive to these types of drugs based on gene expression data. So as an input, they have gene expression data, and they wanted to see if these cell lines are sensitive to the drug they were interested in. They reached out to us for a discovery partnership project, and I will briefly explain what the team has done, but yeah, as I said, the team is here, so if you have some more technical questions, we may try to answer together. So first, how drug sensitivity is measured and predicted. First, if you're interested in how some cell line respond to a drug, usually this cell viability is measured at several increasing doses of that drug. So you will never treat that cell just with one concentration, but you will use several dosages to see what is the optimal, or what is what are the limits when how these cells respond to this drug. You usually compare that treated with untreated control, and based on these responses, there are some summarized metrics that are used for the complete cell line. So in analysis, you are never using this particular one measurement, but you're using these summarized metrics. There are several of them. This is area under drug response curve, area above under response curve, drug response curve, and these IC50 values. Maybe there are more as well. But the thing is that these are summary metrics. And then if you have some representative set of data, for example, you have gene expression data for different cell lines, and you have some of these drug response metrics measured already, you can build a model on top of that data set and try to predict how your samples will react to that drug based on their gene profile. The problem here, there are two problems, at least. So the first one is to choose representative set of data. It is not so easy. I mean, you cannot create it yourself unless you are some huge pharma company or whatever, but still, it is very, very time-consuming and not so easy to create such a data set. This is first problem. And the second problem is what model to use, how to set parameters. There is no best practice. There is no general solution that will work on all data sets in all cases. So this is a playground that we had, and this is something the team could play with and experiment. For a set of data, they used this CTRP portal. So this is a Cancer Therapeutics Response Portal, which have several cancer cell lines with gene profiles and these metrics calculated, summary metrics. And for model, I will speak what models they have used. So this is our solution to the problem. These are the resulting solution which they came up with after several of experiments. So our team proposed to first filter the data, of course, and to clean up the data. And then they also decided to focus not on these summary metric, but instead to use these individual concentrations just to have more data points and to build another variable into the model so they can predict more accurately and precisely. Then they approach feature selection in several ways. After that, they decided to use XGBoost model, and they work a lot with hyperparameters, how to optimize them. And when they find the best combination, they trained the model using these selected features and these optimal parameters. And at the end, they calculated these overall metrics based on these individual ones, because these metrics were the one that a client was interested in. So I will just shortly go through some of the steps without much details. We don't have time for everything. So the data set they started with had 686 samples, but after cleaning, not all entries had gene profiles and not all entries had these AAC measured. Metrics measured, so they did this data cleaning. They also removed some cancer types for which the client wasn't interested, and they were left with 516 samples at the end. Those set of samples have been splitted into proportion of 70, 50, 15, where practically they use for training 361 samples, 77 for validation of models, and 78 for final testing. As I said, they decided not to use these summary metrics, but to use individual doses. Then feature selection was very important here, because initial data set had around 19,000 of features that are genes present in these samples. They have been experimented with different feature selection approaches, and they decided to proceed with first filtering features based on Spearman's rank correlation, then to perform forward selection of features, and additionally to try to narrow down the set of features with backward selection. So they also had opportunity to experiment with different models. They use ElasticNet, generalized additive models, and XGBoost. And for metrics, they used root mean square error and normalized RMSE. And practically of all these combinations, I mean, all these models have been trained with different hyperparameters, with different feature selection options, and XGBoost had outperformed each one of them, so that is why it was selected for the final solution. And at the end, as a result, I cannot share here the concrete genes and things like that, but just to say that final model contained 20 features. And the team additionally did some literature research to understand these genes, these features, to see if they are relevant for this particular case. It turned out that 40 out of these 20 features could be associated with some type of cancer. Then there was one gene that the client was interested in from the beginning, and this gene was present among these 20 features, which was a great result for the client, of course. And one, let's say, side effect was that there was another feature, another gene in the model, which the client wasn't aware of. So they started to explore possibility to use protein coded by that gene as a possible biomarker in liquid biopsy. So that's it for me. Thank you for your attention, and I don't know if you had time. Do we have time for questions now or
Later.
later? Okay, thank you.
Vesna, thank you so much for your participation. Here is a certificate for you. Thank you very much.